in section 3, we're going to look at more of innate, but we're going to start to introduce adaptive as well. Remember that they're not mutually exclusive. So in question number 1, which antibody is known for its role in defense against helminths, so worms? Okay, so let's first of all talk about the antibodies that are out there. We'll get into B cell, plasma cell, and all that, but let's just talk about the general antibodies that you have working in the immune system. So you start off essentially with IgM. Okay, this is a pentameric antibody which is capable of activating the complement proteins, which we'll get into as well. But essentially, this is the first antibody. So let's say that you have some kind of a virus, for example. Let's say you have hepatitis B virus. The first thing that's going to show up in terms of your antibodies will be IgM. And then as you switch into different types of antibodies, then they go from IgM to IgG, okay, to IgE to IgA. And each one of these particular IGs have a specific function. So we talked about IgM is the first on the scene, and eventually you isotype switch, we'll go into in detail, to IgG. So if you've had an infection or you've had, for example, previous exposure to hepatitis B, let's say, for example, I've been vaccinated, okay, with hepatitis B, then you would see that I have hep B IgG, okay? If regard to IgE, IgE is known for, essentially, as you probably know if you have allergies, is it's known for two things. It's known for allergies, okay, and type 1 hypersensitivity, which we'll get into. So in other words, release of binding to mast cells, releasing histamine, hay fever, all these kinds of nuisances. And IgE is also due to helminths, okay? Or not due to helminths, but is instrumental in being able to eliminate helminths. And IgA is a great immunoglobulin. In fact, it's one of the most common immunodeficiencies that is out there. But what IgA is, it's sort of, as was described to me, it's like the immunopaint of your body. So it essentially paints all your mucosal surfaces and gives you immunity from those types of pathogens that try to infect you in a mucosal way. To give you an example, a very important clinical correlate would be with regard to polio. Okay, so polio, the polio virus will try to infect you via your mucosal surfaces. Now, if you have the live polio vaccine, the Sabin vaccine, which we don't do in the United States, but if you have the Sabin vaccine, okay, to get a live vaccine, you will get IgA, okay, because the vaccine touches the mucosal surface, just like some of the live flu influenza vaccines will give you a IgA immunity, and that will give you this IgA paint that will paint your mucosal surfaces. So that's often a differentiating question, is in order to have this IgA, right, then you need to have had a particular live vaccine. As opposed to Salk, okay, Salk killed, okay, you'll still have immunity against polio, but it's not going to be of the IgA mucosal paint variety. So that's just something to bear in mind. Going back to the question, however, where it talks about which antibody is known for its role in defense against helminths, of course the answer is going to be D, IgE. Now IgD we'll talk about later, but it's, you know, we say it's never the right answer. It's not something that is typically, you know, tested or thought about with regard to actually, you know, a role in immunity itself, but we will talk about it later. Question 2, what do eosinophils secrete which is toxic to helminths? Okay, so we have this worm, all right, that's a worm, and IgE, which we said in the previous question, okay, is going to bind to this. So we have an IgE you know, antibody, let's say hypothetically here, and so that's binding to that. And then our little friend, the eosinophil, comes over, okay, this has an FC portion on the antibody, he sees it, This is the eosinophil. Da-da-da. He sees this. He has a what they call an Fc epsilon R receptor. He binds to that. Remember, this is IgE. He binds to that, and then he sparks off, this is our eosinophil, and he sparks off what they call major basic protein, MBP, or ECP, eosinophil cationic protein, as I recall. But the one that they like the best is this major basic protein, and that is toxic to this guy because our phagocytes, look how huge this guy is. One podcast I heard was that it's just too big for a phagocyte to eat, essentially, and so you got to kill him by throwing out all of your toxic reactive species there. So question number two is granzymes. Remember, granzymes are secreted by CD8 cytotoxic T cells as well as NK cells. Lysozyme, remember, that is what chops up that gram-positive cell membrane, and that's in tears, sweat, etc., also in lysosomes. And then the right answer is C, major basic protein. Now, number three is, what is one mechanism by which NK cells interact with the target cell? Okay, so remember how we talked about those IL-7, right? This is IL-7 is the growth factor. We have the stem cell up here. IL-3 is like Switzerland. It can go to myeloid or it can go to lymphoid. And IL-7 is going to produce T cells, B cells, or NK cells. And NK cells are a unique cell, right, which we'll go into in more detail. But essentially, what they do is that a couple of things. Number one is, when they're activated, they secrete a very important cytokine called interferon gamma. Now, we've already talked about interferon gamma earlier because what we know so far is it increases the expression of iNOS, okay? And it also activates macrophages, and it also activates T cells to become I call them the George Bush T cells, okay, because they're very good at eliminating pathogens to sort of kill them all, let God sort them out. Th1 cytokines, which we'll get into in more detail. So NK cells are who secrete, one of the cells that secrete this interferon gamma. Okay, so that's one process that it does, is it's secreting a very critical cytokine. The other thing that NK cells can do is, if there's a pathogen, okay, and that's what this question is dealing with. If there's a pathogen, he's a bad guy, he's got little horns here, and by the way, he happens to have been recognized by an antibody that is bound to here, and you've got this FC portion here, okay? So you have what they call ADCC, antibody-dependent cytotoxic, I think CC, I believe. Anyway, this is one function of an NK cell. So an NK cell also has an Fc gamma R receptor, okay? So this would be an IgG that's lopped onto a bad guy, and then the antibody-dependent killer component of the NK cell is another way that this is eliminated. And NK cells, as you might imagine, are quite good at not only sort of extracellular pathogens that are coming in, but also for cancer cells. So they're nice, and they have a marker. One of their calling card is CD16, which I've never seen tested, but it seems like it comes up a lot in review books. And I think of 16, like the NK cells being sort of a crazy 16-year-old that's out there ready to cause trouble for any bad guys. Okay, so what's the other aspect? Or let's go through this question first. What is one mechanism by which NK cells interact with the target cell, which is what I just said. A is ADCC. B is cell-mediated immunity. No, that's a T cells. C, phagocytosis. They're not a phagocytic cell. And D, endocytosis. They do not endocytose, okay? But they do interact with this ADCC, and they produce these granzymes that in perforins, which basically drill a hole in the bad guy. And you can increase their cytotoxicity, which we'll get into once we get into more cytokines. Number 4. What does the NK cell have on it in order to interact with the target cell? And that is what we just went through. Is the answer is a Fc gamma R. Fc epsilon R, that's E as in IgE, that's what the eosinophil has to interact with your IgE with Helmus, which we just talked about. CR1, that's what a phagocyte has that interacts with complement receptor, which is C3b for opsonization, and then D is CRP binding protein, which also interacts with, in the case of opsonization, which we talked about. Number 5 is, what cells have MHC class 1 molecules on their surface? Okay, so this is a critical component. We'll cover this and then we'll move on. So here's the deal. In terms of immune recognition and antigen presentation. So let's just say your basic cell, any nucleated cell, okay, has what they call this MHC class 1 molecule. And the way that I think about it, it's almost like a flag out there on your cells, and it could be in its little pocket. It could be displaying, let's say, one of your proteins. Or if you had gotten infected by a virus, say, it could be displaying some aspect of that virus, okay, in the pocket. All right, so it could be essentially, well, it's sort of like a kill me signal, right? If you've been infected by a virus, then you got to die because there's nothing we can do for you, and we want to eliminate the ability for the virus to bud out of that cell and go infect another cell. Okay, so that's one of the functions of an MHC class 1 molecule. Again, it's on all nucleated cells. Now let's talk a little bit about that in terms of who has MHC class 1 or how it's produced. So the way that they describe it is essentially that the MHC class 1 molecules are specific to the species. You and I have different MHC class 1 molecules, and in fact that's why I might reject your liver, or if you were to give it to me, or your kidney or something like that, is because we have a different MHC class 1. And these MHC class 1 molecules are antigenic in terms of they stimulate the immune system of non-self. Okay, so that's one aspect of that. And if you look at how these little guys essentially how they work is they call them human leukocyte antigen, and there's A, B, and C, okay? And you have one from your mother and one from your father. So they're co-dominant expression, and co-dominant expression. So when you're looking at, for example, a transplant, clinical correlate here, a transplant match, you try to have a 6 match. In other words, if it's co-dominant, so let's say it's my brother, would be great if he was going to give me one of his kidneys if I had a HLA-6 match, meaning that our A's, B's, and C's matched up with each other. Okay. Now, the other reason why MHC class 1 is important, another clinical correlate, is sometimes there's been some diseases associated with the particular HLA. Okay. For example, ankylosing spondylitis, you'll see it on just about every exam, HLA-B27. There's about a 90%, as I recall, correlate between having an HLA-B27 and having ankylosing spondylitis. That's why they like to test that. There's other things. There's some lupus. There's some type 2 diabetes. There's celiac disease, have these HLA associations, but the strongest correlation is HLA-B27. So remember that in terms of MHC class 1, it's not only just this one, okay? So let's go back, reverse, back to immunology, and with regard to recognizing self from non-self and infected cells and non-infected cells, is that you can increase the expression of MHC class 1, okay? And again, color me surprised, what increases the expression? Cytokines. So interferon, alpha, beta, and gamma all increase the expression of MHC class 1. Okay, so these cytokines, interferon alpha, beta, and gamma, will increase the expression of MHC class 1. And as you might imagine, this is chemistry, okay? So if the particular cell, all right, who recognizes MHC class 1, that's an important distinction, can be recognized by the NK cells, which we just talked about, or by the cytotoxic T cells, okay? So these are the CD8 positive cells, which we haven't talked about, but we will. So either the NK or the CD8 positive T cells can recognize the MHC class 1. And if it's not the right signal, i.e. it's not self, it's not your laundry hanging out there on the line, then these cells have the ability to kill that cell and eliminate the ability, again, of that virus to proliferate, to bud out of the cell and to proliferate. Now let's discuss the difference between, and we'll ask questions about this to reinforce it, but the difference between an MHC class 1 molecule and how an NK cell sees it versus how a CD8 positive T cell sees it. So first of all, okay, this is a virally infected cell. Okay, and he's crying. He's very sad, okay? He has a virus, and there's an MHC class 1, okay? And we had an increase in the expression of interferon alpha and beta and gamma. So he's got a lot of MHC class 1, okay? Along comes a CD8 positive T cell, okay? And he recognizes MHC class 1. And CD4 cells, while we're at it, recognize MHC class 2. And the way to remember that, like everybody says in the review books, is 1 times 8 is equal to 8, and 2 times 4 is equal to 8. Okay, so that's how you remember which one recognizes which we'll go into more of the clinical correlation of that. So as soon as this CD8 sees all these MHC class 1s increase and express there, he's going to kill them. Okay, so an increase in MHC class 1 due to these cytokines secreted in the case of viral infection is when you get these cytokines are going to cause the CD8 positive cell to kill that, to blow him up. He's done, and his virus is done, and he can't bud out, and he can't infect other cells. Now, this is the confusing part, is there's a different response from NK cells, all right? So in the case of an NK cell, this is just happy day. Let's just pretend like, it's a typical cell. He's nucleated. He has an MHC class 1 cell, okay? Now, in this case, so suspend the CD8 positive reaction where you have a bunch of MHC class 1, okay? So in this case, I liken it to when I was in Iraq. For example, I was an embedded journalist with the Marine Corps, and the Marines had in our own unarmored Humvees these little flags, right, with diamonds on the Humvee so that the planes above could see that it was friendly, so they wouldn't blow us up, okay? So in this case, this is how the NK sees it. So he comes up, he sniffs around in the MHC class 1 cell, he sees that it's you, he sees itself, he recognizes self, and he's like, okay, see you later. He gets an inhibitory signal, okay, and he doesn't blow up the cell, all right? It's just like those F-16s and F-15s flying over and A-10s. They're not going to blow me up in my little unarmored Humvee because they see this signal, it's an inhibitory signal, and they move on. Now, let's say that I get captured by, whatever, an Iraqi tank or something, and they rip off this, okay, which is what happens in this case of a virally infected cell. You're virally infected, your normal cell function isn't working, so you can't put your laundry out here in your MHC class 1. In fact, you can't even put the MHC class 1 out there when you have a virus. You're not feeling well, okay, you're sick, you're infected. Oh, in this case, the NK cell's like, what? Wait a second, you know, where's the norm? Where's my little flag that says everything's happy? And then he doesn't see it, and now he's got the attack signal, and kaboom, it blows up, okay? So that's the important thing to remember. In CD8, MHC class 1 is increases killing. In the case of an NK cell, when you don't have MHC class 1 because you have a sick cell, then you may not have the inhibitory signal, and you may end up killing that cell. So that concludes that section of MHC class 1 and 2, and we'll continue with some various ways that bugs can manipulate that tomorrow. Thank you.